So, good morning everyone. I hereby open this academic session in which the candidates Ankit Tanwar will defend the academic thesis Microbiome and its role in shaping planetary health. Welcome.
Thank you, sir.
May I invite you to present us a summary of your thesis?
Thank you so much, highly esteemed Pro-Rector. Good morning to one and all present here. Good morning to all the assessment committee and to my supervisor, to the audience present here on site and online. Good morning to all. Today, I, Ankit Singh Tanwar, will be presenting my PhD Viva presentation titled Microbiome and its role in shaping planetary health. Before we dig deeper into this topic, we need to know little keyword elements about this topic. So, the keywords from this title we could see microbiome, importance and role, and role in planetary health. So microbiome actually refers to the collection of microorganisms, including bacteria, viruses, fungi, and single-cell organisms that inhibit or that a particular environment, such as in a human body. They can also inhibit an environment in a distant ecological niche. And these microorganisms coexist in a dynamic and a complex ecosystem, interacting with each other and with their host. There are several sources for these microbiome, which can be soil, aquatic, animal, and human sources. Now, depending on its sources, it has a different role and applications. In human microbiome, they are essential for digestion, nutrient absorption, skin health, immune system modulation, protection against pathogens. They influence metabolic health, immune functions, and they have potential interventions in therapeutic interventions. So in soil microbiome, they are essential in nutrient cycling, organic matter decomposition. They optimize crop yield, reducing the need for chemical fertilizers, and promote sustainable agriculture. In aquatic microbiome, they are important to maintain healthy aquatic life, nutrient cycling, monitoring and managing water quality assessment, and pollution control. While in industrial microbiome, they're used in processes such as fermentation, biofuel production, or bioproduct synthesis, and they promote a sustainable production, reducing environmental impact and resource consumption. Now, the approach is to dive deeper into this micro-universe or to understand this microbial microbiome. We have few approaches which can be taken with the help of omic studies or microbial genomics. One could be taking comparative genomics, which involves comparison of entire genomes of different organisms. Metagenomics allow us to investigate the composition of microbial community, while metatranscriptomics focuses on what genes are expressed on the entire microbial community, and it shed lights on the functional profile of the microbial community. Metabolomics are comprehensive analysis which all metabolites of a sample are identified and quantified. Now, to give insights on the comparative genomics approach, here I present two of my studies as an example to investigate microbial strains. In the first study, we had compared 3 strains sequenced and categorized for their antimicrobial, cytotoxic, genomic, and proteomic profiles. The selected species was Clostridium tertium, CT, Clostridium butyricum, CB, and Clostridioides difficile, CD. In the study, the strains were named as CT-MALS001, CB-MALS002, and CD-MALS003. While in the second study, 4 strains of S. aureus were isolated from diabetic foot ulcer infections and were compared. The strains were MUF168, 256, MUM270, and MUM475. For both of these studies, the analysis which were run were genome assembly and annotation, whole genome comparison, sequence comparison, pangenome and BUSCO analysis, antibiotic resistance and virulence gene identification, biofilm forming assay, cell cytotoxic assay, variant analysis, and biochemical characterization. Now, the genome sequences of 3 Clostridia species were compared with their reference strain to detect genomic rearrangements. As we can see from this figure, the CB genome, which is highlighted in red, is the strain we sequenced compared with its reference. The blocks represents the identity level. If we see a little gap in between, this could be the lowest dissimilarity regions, and if there is a gap, it is present something like this. In case of CD genome, we could see a lot of genomic rearrangements or shifts That's why the lines compared to the genome references could be seen shifted here. And there is presence of an inversion in the CD genome. While in CT genome, we see a lot of dissimilarity compared to the references, a lot of shifts and few inversions present in this genome. Genome annotation plays an important role in the field of functional genomics. It depicts the functions of genes and other genomic elements. In these three strains, we could see that the CD genome had a high number of percent abundance gene in phases and transposable elements. In case of membrane transport, CD genome had a very less percentage of gene abundance, while in case of motility, CB and CD had a very high number of percentage compared to the CT genome. While in case of metabolism of aromatic compounds, CD had dominated the category compared to CB and CD, while nitrogen and phosphorus metabolism, CD genome had a very less percentage of genes involved in this category. In pan-genome analysis, we could see CD genome shared more than 70% of its gene core with its references, while CB genome shared only 57% of its core genome with its references. And CT shared only 47% of its gene in core category. We could see CT MALS001 had the highest number of accessory genes that could also correlate with the high category of miscellaneous gene in genome annotation step. In antimicrobial resistance and virulence gene, in this figure, we see genomic coordinates of antimicrobial resistance genes and virulence genes identified in all three Clostridia species. We could clearly see here the presence of 31 gene cluster flagellal proteins which codes for CD motility. And we could also see presence of main exotoxins tox A and tox B along with its binary toxin CDTA and B. Along in the terms of antibiotic resistant gene, we can see an island of VAN genes which confers resistance to vancomycin antibiotic. In case of CB, there was a presence of a exotoxin PFO-A under core toxin category, and for CT genome, there was presence of a NACK exoenzyme in the core toxin category. Biochemical characterization showed that CB and CT were able to hydrolyze starch, while CD did not, because CB and CT had presence of alpha amylase and oligo-1,6 glucosidase enzymes which could hydrolyze starch, while CD had lacked both of these enzymes. In case of motility, we could again see that the CD had a high swimming motility and was diffused growth rate because of the presence of the flagellate proteins which we saw in the earlier image. While CB had little less compared to the CD, while CT lacked a lot of flagellate protein, so that's why the motility was less. In biofilm forming assay, both at 24 hours and 48 hours, we could see the dual cultures of all these species were higher compared to the monocultures. As we can see, significant higher biofilm production compared to monocultures. In cell cytotoxic assay, the percentage of cell survivor decreased as after 24 hours, 48 hours, and 72 hours significantly compared to the control, which we've seen for all the three strains. Now, in the second study, where we compared 4 strains of Staphylococcus aureus, we saw 14 antibiotic resistance genes were common between all the 4 strains, and 56 toxin genes were common between all 4 strains. Antibiotic resistant gene profiling here shows the genomic coordinates of all those antibiotic resistance genes, and to confirm these presence by molecular methods such as PCR, we could see as in the figure for MEC-A, lane 4 and lane 5 represents MUM270 and 475. We can see clearly 2 bands present and the presence of MEC-A through in silico analysis for MUM270 and 475. We could see the same for ANT4, a presence in MUM270, and ERMC also present in MUM270 genome. Virulence regulatory network depict here for Staphylococcus aureus shows all the important virulence factors involved and how they are positively and negatively regulated. It shows few of the major exotoxins of Staphylococcus aureus, which are HLA-B, SPA, and SCB. We could also see the protein, blast protein identity, and in all the 4 strains, we could see that enterotoxin and pore-forming toxins had the highest protein identity for strain MUF 256. In the second approach, one can take its metagenomics. I analyzed metagenomic sequences of 272 healthy volunteers classified based on Prakriti, Kapha, Vata, and Pitta. Coming from an Indian background, I decided to classify the samples based on Ayurvedic medicine, and the combination of the 3 doshas help in determining the Prakriti of an individual, and any disturbance in these equilibrium can lead to Vikruti or a disease condition. The stool and saliva samples for all the 272 healthy volunteers were collected, followed by metagenome sequencing, data analysis and visualization. And the data analysis consists some concepts like raw data and processing, microbiome diversity analysis, detection of microbial enterotypes, core microbiome analysis in gut and oral microbiomes, and identification of Prakriti specific signatures. In terms of average relative abundance at phylum level, we could see in gut microbiome for all the 3 classification between gender male and female, Bacteroidetes had a higher abundance compared to other phylums, followed by Firmicutes. While in case of oral microbiome, Firmicutes and Proteobacteria had a similar percentage abundance, followed by Bacteroidetes. While in terms of alpha diversity, between the 3 classification, the Vata samples had a high alpha diversity in gut microbiome samples, while in oral microbiome samples, Kapha and Pitta had similar biodiversity, microbial diversity, followed by Vata. Most abundant OTUs at genus level for gut microbiome were Prevotella and Bacteroidetes, followed by Dilester, Roseburia, and others. While for oral microbiome, it was Streptococcus, Neisseria, Veneola, Haemophilus, Prophomonas, Prevotella, and others. Between the 3 classification in gut microbiome, 91 genuses were shared, and we could see on the basis of 2 abundant genuses that the 2 clusters were formed between these 2 abundant genuses. While in oral microbiome, between the 3 classification, 99 genuses were shared, and there were no clear clusters formed on the basis of OTU abundance in genus level. There were some overlapping clusters. Now, on the basis of phenotypic classification at phylum level, in gut microbiome, we could see mobile elements, biofilm former, stress tolerant, and potential pathogen. All these 4 categories were dominated by only one phylum, which was Proteobacteria. While in oral microbiome, all these 4 categories were actually a mix of different phylums present in them. Now for the summary and confusion part. The summary, comparative genome analysis identifies genes and regulatory elements. It unveils genetic mechanism, differences, and evolutionary history, aiding in functional inferences. Now, comparative genome analysis between pathogenic and non-pathogenic strains can help identify genes and factors linked to virulence. These methodologies enable identification of emerging pathogens. Metagenomic analysis help understand the dynamics of a host microbiome relationship. It is widely used to study the microbial communities, and the taxonomic classification can shed a light on type of microbes present in the samples. Metagenomic analysis also can help identifying pathogen in clinical samples, aiding in the diagnosis of infectious disease, and composition of certain microbial communities can act as markers to describe a disease state. Now the confusion part, what all this has to do with planetary health. So, to answer this with this graphical abstract, human microbiome associated health data collected at any level, whether it's personalized, community, or global health, can be used for pathogen screening, antimicrobial screening, for studying different microbiomes present in human, or checking the relationship between a disease and a microbiome. All these data can be used for genomic surveillance purposes to monitor emerging pathogens, infections, or emerging disease and pandemics. When this data deposited in global science platforms, the researchers around the globe can utilize this data to have the applications in the field of planetary health. With this, thank everybody for listening, and I now give the session back to Pro-Rector.
Thank you very much for a somewhat technical but very enthusiastic talk. And you showed that in the end everything hangs together, which is kind of nice. So before we start with the opposition, I would like to introduce the supervisory team, because you will probably wonder why two people will not ask anything, but they supervise the candidate and they will not ask any questions anymore. So that's Professor Dr. Angela Brandt from Public Health Genomics, welcome. Professor Satya Murthy from the Department of Genomics from University in Dharwad, India. Welcome. We will start with the opposition, and the opposition will be started by Professor Dr. Kasia Chabanovska. She's from the Department of Public Health of Maastricht University. She acted as chair of this assessment committee, and she's at the moment also secretary of this committee. The floor is yours.
Thank you very much, dear Pro-Rector. And I would like to turn to the candidate and the supervisory team and congratulate you on the thesis, which is very important and very interesting and has stirred a lot of thoughts when we think about what lives in us, on us, and how it is related to the planetary health, and also what impact it has on soil, animals, and food. So thinking about that and diversity and interconnectedness and the research that has recently grown exponentially in relation to microbiome, I would like to ask you about certain considerations that you must have taken while collecting the biomass samples for the studies. What were the specific considerations that you took in your 3 studies? Yes, that's a great variety, I understand. And what were the caveats? And how you addressed them?
Thank you so much for your question, highly esteemed opponent. For this study, comparative genomic study, which we had for 3 strains, the main criteria to select those 3 strains, which were Clostridium Clostridioides difficile, which is a known pathogen in CDI infections, while Butyricum was selected because of its dual role as a probiotic plus as a pathogen, and CT, which is in most cases known as an unknown or an emerging pathogen, was selected. So on the aim of the study was to compare between these strains and try to get a conclusion that if CD is actually an emerging pathogen or not. For the second study, in the Staphylococcus study, Staphylococcus is a more prevalent organism in diabetic foot ulcers. So we had 2 strains which were collected from the female samples and 2 from the male samples. And between, other than the gender, there was nothing different parameters taken in this study. But
like culture neutral or diet neutral samples? Was this?
No, so these samples for the Staphylococcus were directly taken from the diabetic foot ulcer wound. So it is considered that when the presence of Staphylococcus is in the wound, it is not able to heal properly. So directly the wound samples were taken, and the majority of the organisms present in that were taken into account. One of the most known pathogen was Staphylococcus, so we wanted to see how the virulence of this strain, even present very closely in the same strain, how does these virulence factors which changes on a minute level can affect the wound healing process or can increase the virulence of the strains. While in the cases of metagenomic study, we had 3 classification which I told on the basis of Prakriti, Kapha, Vata, Pitta. Now, Prakriti actually serve as a personalized medicine, because we are actually distinguished between these samples as 3 categories on the basis of some characteristics. It can be weight, it can be height, it can be color of the hair, or like BMI, so and the gender. So few of these things, what kind of diet? Diet was not included in this study, but it could be one of the factors. So now depending on these factors, you can divide them into these classification, and maybe the more factors you include in this study, maybe you will get more Prakriti classifications. So there are actually 7 Prakriti classification. The factors we had taken, parameters we had taken in this study had only could only classify them in Khava, Vata and Pitta Prakriti. But if you take more parameters into this, you can actually define them into a lot more Prakritis compared to this one.
Thank you very much for the answer.
Yeah.
And then we move to the second question.
Yes.
And that relates to this big thinking about cloud and data and global ownership of the data.
Yes.
And I believe that there must be a lot of ethical considerations. So what are some of the ethical aspects that need to be considered when we talk about the microbiome research?
Yes.
Ownership, for instance, and not doing harm, and justice, and some other aspects. What do you think about ethics?
Yeah. Thank you so much for your question, highly esteemed opponent. That's for sure that on a study on a global level would for sure have security threats or ethical threats, like, because if we collect one study from a demographic area, put it on a cloud server, and a region from somewhere else can use or get to know more about that population and can maybe use it for, in a harm way. So it is important to define few guidelines for what kind of data has to be uploaded, how much it has to be anonymous, and how much has to be go for somebody on the online to analyze it, or some other researchers to analyze it for. So these guidelines would be, first has to protect the individual, then it has to be on a level where this data cannot be used in a way to harm the population which the study is going for. And these guidelines has to be set by some regulatory or policy makers, which in government, or to protect the citizens of a demographic area or a region from all these threats.
Are you aware of any guidelines, or is it in the development?
There are cloud platforms, but currently the amount of data they upload is either the consent is taken by the patients, from the patients, while the hospitals collect that and put it on the cloud servers. And the data, the metadata which they collect, it's not that much that it can harm any kind of population till now. But later, if we are putting that more and more number of metadata or more type of classification or data has to be put on server, it is important to put some policies over it to regulate the data into these cloud servers, especially considering all the security threats of hacking or leaking or using maybe these data for biowars can be done.
Thank you. Yeah. Thank you very much. Yeah. And my last question relates to chapter number 3. Yeah. Yes, Cancer Insights, and this is a kind of review.
Yes.
I have a question, how this review was performed, especially how the specific categories which are discussed were identified. That's chapter number 3, Cancer Insights, a pathway and network analysis.
Yeah, the categories?
The titles, the categories that are described in the review.
Okay.
Because so it is a review, and how you came up with these areas, and how the studies were selected for this review.
So first thing was, when we were studying about this microbiome, we had to associate it with one of a major public health issue. And cancer is a global burden, as we all know it. So microbiome around the world has been studied all over around, that it's associated with gut microbiome and cancer. Some have a beneficial role, some actually worsen the condition of a tumor-associated associated microbiome. So when we were introducing cancer into this term, we, as for the second chapter, we could see that oral and gut microbiome was defined. So we could also show a major burden in oral cancer, and how this oral cancer can be taken in account to study the tumor environment for, as an example, to actually bring down a therapeutic intervention for oral cancer. So we wanted to connect the microbiome to oral cancer or to the cancer burden so that it can be used as a therapeutic intervention for that.
Thank you very much, and I thank you very much for your answers, and I turn back to the prorector. Thank you.
Thank you.
Thank you very much. The opposition will now be continued by Dr. Bier. He has a chair in genomics. He is from the University of Potsdam, Germany. I'd like to invite him to ask questions, and welcome to Maastricht.
Yeah, thank you very much for the invitation, Mr. Prorektor, and thank you for this introduction to this interesting work and your talk about the microbiome and its role in shaping planetary health. So it's a real global approach. And I want to, since my background is diagnostics, I go a little bit to the starting point, to the data. And my first question is going to the genomics of the antimicrobial resistance. So how is the microbiome analysis linked to the organism? So how important is it to know which kind of organism, of species, are involved in this special resistance scenery?
Thank you so much, highly esteemed opponent, for your comments and reflection on the thesis. If these organisms which are selected on the basis, like how to check the pathogenicity of these organisms can be checked on the basis of experimental methods, can be checked through in silico analysis also. When these pathogens were selected based on few of the major global burden like cancer or CDI is a very common infections present in hospital settings, and Staphylococcus in diabetic foot ulcer. So diabetes is a very common burden, and it is been linked that these presence of these microorganisms actually worsen the condition. So based on these settings, we had identified the pathogens and took a genomics approach to understand at a micro level that how the difference in the virulence factors present in that can actually act as a burden, can change its impact. And antimicrobial resistance also again is a global AMR burden. So AMR was studied for this. Because if the pathogens are resistant to all these resistant for this, there might be a reason. Either there are horizontal gene transfer presence in between, or there are some other factors which can be maybe presence co-occurring pathogens presence in the microenvironment, which actually transfers the virulence capability or the AMR property from one organism to another organism. Now, plasmids also play a very important role here, but we did not go in the plasmid studies for these this thesis. But plasmids include genetic transfers of these mechanisms which actually provides virulence to the other organism present in the same culture area or in the same micro environment. So, I hope.
Yeah, thank you.
Yeah.
Thank you for this answer to connect these topics. And I want to go ahead in the genomics of the disease. There's a host-pathogen interaction. So you are looking now to the microbiome and the genomics of the different pathogens involved. what would be the next step looking to the human genome, correlate the human genome to the pathogens? Is there some relation or is it completely different? So this is my question.
Thank you so much, highly esteemed opponent, for your question. The next step would be, the whole point of this thesis was to focus on these microorganisms as emerging pathogens. Now, the already existing information can be taken to have a surveillance system on the coming microorganisms or viruses, where we can check when their patterns of AMR changes or virulence changes. When we have a kind of a timeline or a framework where we can see that if the virulence of the AMR burden is changing in these microorganisms, they can act as a threat. So these microorganisms are studied again at genomics level, that what are these changes or mechanisms involved which are actually contributing to its virulence or resistance. So the next steps on this is to have, as I said, in a global scale, if this information, put it in a cloud server where we know from which region we had a resistant strain, and from which what is the actually AMR pattern going on around the world for one species, we could see that how that pathogen or the organism is becoming pathogen on the over time. So genomic surveillance is something we are looking here using the cloud servers, if the data has been put on the cloud servers from these genomic studies, multi-omic studies. Yeah.
Yeah, but my question goes in the direction, can you link this in future maybe? It's not, I mean, just starting with your thesis, and now I'm thinking ahead with the patient's genome. Is there some correlation?
Yes,
because
yes.
It's you can see, look at it as an interaction of genomes, so to say. Yeah. And then you would need other information. You think you can get this out of the cloud?
Yeah. So, as in case of personalized genomics, microbiome has a very different relationship with its host in different individuals. So maybe taking the personalized genomics approach, one can actually see what kind of presence of organisms can be actually helping a beneficial role in the patient, and which one can actually be threatening to the individual. So if we know that there is a community of microbes which can prevent the spread of infection or actually hold or kill the other bacteria, we can actually transfer these kind of, we can transfer it by probiotics or maybe fecal microbiome transport, where you can actually stabilize the system of microbiome in a patient in cases of stomach cancers and other colorectal cancers and all. This can be correlated at that stage. So we can use one beneficial microbiome to actually kill another pathogen.
Okay, thank you. Interesting. Then I will go to my last question and even broaden the scope again to looking for genome-environment interactions. So how can you introduce environmental factors as well into the analysis? So is there some impact to the genome, or maybe it's not the genome?
Thank you for your question, sir. Sure, epigenetic factors or environmental factors do impact genome of an individual, and they will have effect on a microbiome also, as stress, diet, or a lifestyle, particular lifestyle, do have actually they have impact on a microbiome inside a human. So these changes also has to be taken in account for the future studies when we are actually comparing these pathogens. Do they only get affected by the genetics factor of the individual, or do they have impact of epigenetic factors also? So yes, these factors in futures can be read, but here as we taken only the comparative genomics to just see the underlying mechanism of it, but epigenetic factors do also have a large effect on the genomics of genetics of an individual, and it can change microbiome time to time. Even changing the demographic region, the diet can change microbiome of a person. So these epigenetic factors do play a very vital role in these changing microbiome genetics mechanisms.
Yeah, thank you very much
Yeah,
for
thank you.
these answers, and I go back to the prorector.
Thank you very much. The opposition will now be continued by Dr. Peter. He's also a specialty in genomics, and he's from the Fraunhofer Institute for Cell Therapy and Immunology from Germany. Welcome very much to Maastricht. The floor is yours.
Thank you very much, and thank you very much for the invitation. Since my background is in the area of biology, and especially over the last almost 20 years now in antimicrobial resistance, I will ask a few questions regarding that area. The first question from my side is, what challenges did you face, first of all, in choosing the different methods in detecting antimicrobial resistances? There are, on the one hand side, the classical microbiological methods, and then on the other hand side, you have the different molecular biological methods. And which challenges did you face in choosing those methods and in correlating the data getting from all these different methods, and were they highly correlated, or did you actually have difficulties correlating them, and how did you decide if there were discrepancies, what the reasons for those discrepancies could have been?
Thank you so much, highly esteemed opponent, for your question. Yes, we had few difficulties. One first, the major difficulty we had in few of these organisms was at the genomic level, we had identified a lot of AMR resistance genes. But when we are correlating that with the phenotypic characteristics, it always never shows, sometimes for few of the genes, it never shows the antibiotic resistance in the phenotypic characteristics or in the lab culture settings. Now, either these genes which are already present in the genome of that strain maybe are dormant and not expressed, or they'll be only expressed in the when provided by an external threat, or in a stress response they could be expressed. So in the chapter 4th, where Clostridial species were compared, in Clostriditis difficile, we had a presence of vancomycin genes, and it correlated with the phenotypic relationship also, because CD was tested positive or resistant against vancomycin antibiotic. While in case of Staphylococcus, one of the strain actually tested sensitive for 9 antibiotics, but it had genes for all those antibiotics. So maybe the characters on a lab and maybe inside an individual are different. So there are factors involved which may be activating it or maybe deactivating it, that it doesn't show its resistance on maybe a lab culture, but maybe in an individual. So these factors has to be explored, whether it is only when a stress is involved that they express these genes, or maybe it is when there is presence of other pathogens that they express these kind of antibiotic resistance genes. So correlating the resistance with the presence of in silico data is always little difficult, but again it is said that just because an antibiotic resistance genes is present in a pathogen or an organism, it doesn't mean it will shortly show resistance against that antibiotic. So yeah, these were few difficulties.
Thank you very much. So this leads to my second question. It is known that just the mere presence of a gene is often not the only factor that is causing a resistance, but especially in the area of beta-lactam resistances, it is known that a lot of single point mutations, SNPs, within the genes are responsible for resistances. There are huge databases that list all the point mutations that are known. They are constantly increasing due to environmental mutational factors, but there are large databases that include these point mutations. Did you also incorporate that in your studies? Did you look only at the genes, like using primers, for example, or did you also go inside the genes and or will you do this maybe in the future? Is this something you would like to incorporate, analyzing the point mutations, which are very relevant, rendering a lot of genes sensitive, resistance genes sensitive again? So often this is something that might be the reason why you have a discrepancy between phenotypical and genotypical data.
I totally agree. Highly esteemed opponent on this. And for the Staphylococcus study, we had a variant analysis, but we did look for the targets of the AMR genes and the variants present which can actually cause suboptimal binding of drug targets or the drugs. But in case of only AMR genes, the variants were not checked. If there was some point mutation which actually causing that to show the resistance or the sensitivity against that. But yes, the study would be, the next steps could be taken into this account, that these point mutations to be considered in these genes to actually look the resistance pattern. But again, limited to the variant analysis, only on the drug binding sites, we had variants which will cause drugs to actually not bind and be not effective. So that was included in the study. But these can be, yes, future steps to be included at both Clostridia and Staphylococcus studies to check for those point mutation, that why these phenotypic characters are not shown even with the presence of the genes inside the genomic coordinates. So, yeah.
Thank you very much. Coming to my last question, I'm moving a little bit to an area that I'm not familiar with so much. It's about the treatments of Ayurveda practice. And so what treatments were used, and did you also check if there, I noticed in studies that there could be a bias often in treatment options. During a PhD, of course, you have only a few years, and if certain treatments were done, let's say, in the summertime, and other treatments were done in the winter, there could be a bias just based on the food you eat and the temperatures, the seasons. And did you take into account those biases, that there's not a misleading information based on those treatments done at different times of the year?
Thank you so much for your question, highly esteemed opponent. So when we were analyzing the samples from these 272 healthy individuals, the criteria which we had set is that none of those samples will be taken in account which had been exposed to smoking, alcohol, consumption, any of these factors. Also, along with that, if they had been taken an antibiotic in last 6 months, they were excluded from the studies. So if it was past than 6 months, then it would be included in the study. So these factors were excluded from all this. And on the basis of bias, which is mostly all these 272 samples were from same demographic regions. There were gender differences, and we had also taken into account ages of all the samples. Along with that, the bias, because we have not taken the diet as an factor in it, so I would say on basis of diet there is no bias, because that account was not taken into in this classification. But yes, if we do classify them based on diet, there might be further classification between these groups. And yes, so the lifestyle, socioeconomic impact, and the diet were actually taken out of from the account before selecting these samples for these studies.
And seasonal differences are maybe not so huge in India because you don't really have a
Yes,
winter.
seasonal differences would not be there because it's not that these samples were collected on a very long period of time, and the region which we had selected, the samples were from Bangalore, so there is not highly changing weather. So it's not extreme winters or summer in these seasons. So I think these season as an outer trait or the characteristics won't be affecting that much to the sample's microbiome as such.
Right.
Yeah.
Thank you very much
Yeah.
for your answers.
Thanks.
And I think then I'm done with my
Thanks.
questions for now. Thank you.
Thank you, Dr. Peter, for the questions. And it's a disappointment that there's not much change in the weather in India. Okay, we continue the opposition with Dr. Eisen. He's from the Departments of Bioinformatics and Neuropsychiatry from Maastricht University. The floor is yours.
Thank you, Prorector. Let me first of all congratulate you and also the supervisory team with the completion of this very nice thesis. And I think what is very interesting to see and very good to see is that it touches upon various aspects. You know, there is wet lab work, there's data analysis, but there is also thoughts about how we implement things in the world and how does it relate to the broader system. I think this is hugely relevant, and I value that you took the various angles where some of them probably were a bit less familiar to you than others, right? So I also like that, and you will see that my questions, if the prorector lets me, let's see, I go a bit across the various chapters on various types of topics. Let me take the order of the thesis a bit, and starting on chapter 2, where we have been discussing already a bit on the dosha and the prakriti. So what I found interesting is that you said yourself in introduction that it's rarely seen that people are very specific of one type. But when I then look at your study, how you had a cutoff of 70% to assign them, that there was sort of seemed to be no one that was not fitting in one class. And I was wondering, was this because of selections you did, or was this a special population or so? So can you elaborate on how you can explain this seeming contradiction?
Thank you so much, esteemed opponent, for your reflection on my thesis and for your question. Now, when this classification was done, as in the thesis, we can see that 70% of one characteristics and 30% mixture from the other two characters was taken into one dominant Prakriti type. So if we add more parameters, now the parameters we had added was gender, age, and BMI into the, while classifying these samples. While in the Prakriti classifications, if the more parameters been added, the classification goes much defined way. So if we add maybe diet into the classification, height, weight as added by BMI, but then also the demographic areas, the epigenetic factors, or maybe the phenotypic characteristics of a person. The more parameters added into that, maybe it will classify into more categories. So it won't be definitely in one dosha, it won't be maybe in kapha, it will be kapha, vata, it will be vata, pitta. So it will classify more into while adding more parameters. So it all depends while selecting the samples, how many characteristics by the Ayurvedic surgeon or the senior has been taken to actually classify it between one of these doshas. So as we had very few parameters, it could actually lie in between one of these classifications.
Yeah, thank you. That's very clear. So it depends a bit on this granularity of the characterization, right? I still want to get back to this diet thing a bit. So I was already addressed with two of the other opponents, and I was still a bit surprised that you didn't take it, because you say yourself, well, of course, the diet is important. We know that, but especially for microbiome, and you even see, that two of the main bacterial families you saw, very much relate to Western versus more vegetarian diets even, right? And you could even think that maybe diet explains everything you see, right? So I wanted to ask you to reflect a bit on, the confidence in your results without taking diet.
So Thank you so much for your question, esteemed opponent. The main reason we didn't put diet because of the variety that it was very hard to get a strictly vegetarian or a non-vegetarian diet. There is a lot of people in this study which were involved had a very mixed type of diet into this. And as it's a gut microbiome study, diet would have added more parameters and more information into this. This diet actually taken out because Prakriti was our main focus. So the personalized medicine based on these 3 doshas was something our focus was. So diet for oral and gut microbiome, in this, as I said, there was a lot of variety between these samples and there was no strictly strict vegetarians or non-vegetarians present in our samples. So the diet was excluded for more parameters, and it was only classified based on the gender or the BMI or based on age factors like that. Just for a very early Prakriti classification study, we had thought to remove diet or maybe add it later in the coming studies.
Yeah. So as a very, very short follow-up, is it known from other studies whether doshas and diet relate or not?
Yes. So in gut microbiome, obviously, the diet makes a very important factor. So it is seen that if it is a non-vegetarian diet, that Bacteroidetes phylum is presents more. While in a vegetarian diet, Prevotella is more abundant. Both of these phylums were actually abundant in our gut microbiome samples. But we now cannot classify them based on their diets. But we do see 2 different clusters which are present on base of the abundance of these phylum. So maybe they also can be classified based on diet if we had that metadata present in this study.
Yeah, thank you very much. Then let's also move on a bit more to the antimicrobial resistance. So following up from the previous opponent, but taking the other angle and connecting these works in chapters 4 and 5 to the chapter 6, where you propose a workflow, right?
Yes.
And you put quite an importance on genomics, to which you would agree. Of course, this is not the question, but the thing is, you also saw this discrepancy between the genomic features and the real resistance of the strains. And I was wondering whether then looking at that would not exclude too many antibiotics, for example, that the pathogens wouldn't really be resistant for. So can you reflect on this?
Yeah. Thank you so much for your question, dear opponent. Yes, these factors, of course, when we are looking at antimicrobial resistance, it's not always only the presence of the genes. We have to look how in that environment microbiome is either maybe threatened by some other stress or the mechanism where it can borrow the enzymatic properties from other microorganisms. So the genomic approaches might be not completely the way to go for it, but yeah, they do pave a path for us to go for the phenotypic studies and understand the factors which can be involving maybe in the pathways of these virulence factors or maybe in the antibiotic resistance mechanisms. That is it the outside factors or only the presence of genetic AMR genes which could actually lead for the resistance studies. And in the chapter 6, as we, the chapter 6 had an aim to outline all the tools and databases present for studying these kind of study, study the virulence and antibiotic resistance studies, where all the resources are present and how one can actually pursue to start with these studies from a genomic level, if they have a genomic data sets.
Yeah, thank you. Yeah. And to connect that in the chapters 4 and 5, when you take the samples and you check whether the strains are resistant, and you often see even when genes there maybe not be, right? But they also may not have been exposed too much. So what is the effect of the timing of when you take the samples for these studies, right?
Yes, thank you so much for your question. Yes, the time when we first culture a microbiome and see its resistance, and the pattern could go down over the time, depending on the environment it has been exposed to. So it can be possible that when we first tested for a strain would be resistant, but later on it has loses its resistant property, maybe depending on where it was exposed to. But it can still gain its same properties because it's encoded, it's in genome. So maybe we present the same stress environment again, it can have the same phenotypic characteristics which we can see on the later stages.
Yeah, clear. Thank you. So then this takes me also to another domain yet, the last chapters. And yeah, it was interesting to see that you reflect on these community-based systems and cloud-based systems and so on and so on. You also refer look at my question, to some of the European initiatives like Destiny and so on. So you talk about twin models, cloud systems, but what I didn't see so much being discussed is how these different initiatives already interact with Destiny. You know, they describe their purposes, but it's not really about infectious disease surveillance or so, right? They don't really include it. And with the cloud systems, they make some suggestions, but don't, again, really explicitly connect to the European Open Science Cloud or so. And I was wondering, are you aware or do you foresee ways how these actually already collaborate even nowadays or maybe in future? How can we, you know, combine your last two chapters into an integrated way to really do this very well?
Thank you so much for your question, esteemed opponent. Now, the already based cloud system are setting up an example that this can be done. So if it is already involved, the system is already present where people are actually collaborating or sharing data between the platforms, that same the same parameters or the same model can be taken into account to also set up a global health cloud where data can be of heterogeneous data. It can be of infectious diseases, it can be AMR, it can be virulence, it can be maybe cancer samples, and there will be security threats and a lot of difficulties at every level of setting up of these systems and collaborating at a different scale. But if we start from a very small level at a very good regulation and policy making, we can set up these guidelines and we can have a system where these approaches can run smoothly. So it is something like once we set up, we face the difficulties, we try for the next solution. Then we have again one more problem, we try to solve that. So it is not easy to just set up together, but yes, it will be.
You may choose to finish your answer or you may forever be silent.
Just with an example, sir. If an app is launched, we have a lot of bugs in it. So once we follow debug 1, we actually become to a stable version of an app. Yeah. So that's how we face one problem at a time, we solve it, and then reach to this global system at the end.
Yes. Thank you very much.
Okay. Thank you, Dr. Eisen. Dear candidate, the time appointed for defending your thesis has passed. You have seen that, right? The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await our verdict here in this room.
Thank you so much.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If there's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Ankit Tanwar, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict, and taking into account your previous qualifications, the degree committee here present has decided to grant you the degree of doctor. Professor Brandt is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Ankit Singh Tanwar, the degree of doctor and grant you all rights attached by the custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Dear esteemed doctor, dear Dr. Ankit Singh, Dr. Singh Tanwar, sorry. Dearest Ankit, our cordially congratulations. So, on behalf of the Satya as well, Satya Sir, Professor Satya Morsi. You have chosen the topic of the public health implications of the microbiome and a complex and challenging topic. Chapeau. Your research has not only expanded the frontiers of science, but also provided crucial insights into the stewardship of planetary health. By analyzing the genetic blueprints of microbes, your work has shed light on how these minute entities play a major role in ecosystems. Microbes, often unseen and overlooked, are now understood as key players in public health. You also added a new ingredient into the microbiome masala research, the Ayurveda Prakriti, the Indian system of medicine which relies on the phenotyping of individuals into the predominantly three conditions like you explained nicely, Kapha, Pitta and Vata. You are indeed an ambassador who bridges between disciplines and traditions. I hope that you will continue this research pathway and that you will add more insights into these important fields. Please, dear Ankit, sail on. Thanks to you, dear Satya, Ankit is my first PhD from India. And not only this, from the very beginning, he has been a research scholar very close to my heart, being always in good mood, being always charming, being always caring, being always on time, except on his wedding. Ankit is a truly gentleman of his generation. Dear Ankit, when thinking of you, I think of a bright color. By the way, it has nothing to do with the fact that you are colorblind. What a challenge in such a research. So when thinking of you, I think of the bright color royal blue. Yes. Royal blue is a bold and vivid shade, often linked with confidence and authority. A person whose personality is described as a royal blue might be seen as a natural leader, someone who commands respect and isn't afraid to take charge in situations. You, dear Ankit, have, for example, always been cutting the birthday cakes of the Manipal School of Life Sciences. Royal blue is also associated with trust and reliability, similar to how banks and business often use blue in their logos to convey trustworthiness. A royal blue personality could be seen as dependable, someone who others rely on and trust. Dear Ankit, we always trusted your advice on best food in town, and I always relied on your fixing Angla's computer. Yes. Royal blue is frequently linked to intellect and wisdom. A royal blue personality might indicate a person who is thoughtful, reflective, and values intelligence. They might be considered as someone who thinks deeply about things and takes an analytical approach to problem solving. Dear Ankit, the key word here is country in, and long philosophical discussions about God and the world, such as, can grasshoppers be colorblind? Despite its richness, royal blue also has a calming effect. It can indicate a personality that is balanced, stable, and able to remain calm under pressure. This person might be seen as a soothing presence in stressful situations. The eligibility stress test for becoming a PhD candidate, the chameleon origami, yes. The eagle origami, you proved to be eligible, being flexible and focused, and here we are today celebrating your PhD. And the term royal in blue brings to mind nobility and dignity. This personality type could be associated with someone who carries themselves with grace and has a dignified presence. Dear Priya, you are lucky. You are married to a yes-man gentleman. Royal blue can be inspiring, symbolizing high ideals and aspirations. A person with this personality might be seen as someone who inspires others and aims high, both for themselves and for the greater good. Dear Ankit, chapter 8 of your thesis, your idea of the global health data cloud, laying new directions for collaborative science, is certainly inspiring. Remember, dear Ankit, color psychology is not exact science, and these associations can vary across the life course of a person. If you are too perfect, dear Ankit, God and Satya will be angry. Let me include your second supervisor, Satya, so in my final words again. Always grow and change for impact and fulfillment. Our PhD journey was such a pleasure, professionally and personally. Thank you, dear Ankit. What will be the next steps in your life and in your career? I don't know, but for sure, they will be full of passion, creativity, curiosity, and they certainly will be royal blue. Again, cordially, congratulations to you, dear Ankit.
Dear Doctor, congratulations.
Thank you, sir.
Also on behalf of this university, we are very proud that you are now an alumnus of this university. You defended very well. You had a very beautiful thesis. I even have now more respect looking at all the colorful pictures in the thesis, knowing that you're colorblind. We will discuss in the drinks afterwards what you really see. I really liked also your front page, in which you have the combination between Earth or Planet 2, perhaps, and the Petri dish. So fantastic, a great achievement. I hope you enjoyed the day. We will now all go to the stairs. There's a big staircase if you come in the building, and we take a picture there, and then we go for drinks afterwards. So with this, I close this ceremony.